Hello guys, I am Shubham from Shubham Tutorials. This video is about double strand break repair pathways. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. So what is double strand break? Basically, both the strands in the DNA causes a break like this. So this is a double strand break. Okay, and this can be caused because of radiation. Radiation or chemical agents that may induce this DSBs. Okay, so these are repaired by the two pathways. First is the homologous repair, and the second is a non-homologous repair. What is this homologous and non-homologous? Basically, homologous in which template is there, right, for the synthesis of a DNA. Homologous repair is done by the recombination. Homologous recombination. So this recombination we will discuss in the separate video when we will talk about homologous recombination, right? In this video we are seeing the non-homologous repair pathway, that is non-homologous end joining. Denoted by NHED, non-homologous end joining. So here in homologous, it is a template dependent. Template dependent. Here there is no template. Right? How your template is dependent? Just we will see a basics. This is the double strand. Okay, strand which is having break, and another strand which is proper. Now what happens? This damaged strand uses this another strand as a template. So this will be template in this pathway. Okay, so this process we will discuss detail in the recombination video. Right. So this non-homologous end joining is what basically there is no template here. Non-homologous end joining. So from the name only we can see that this is non-homologous pathway. Right. So the sequence, original sequence is lost. Okay, as there is a double strand break and the original sequence is not restored. Very important. The original sequence is not restored. So this process is mutagenic. We have to remember this. This is a mutagenic, means the mutations are produced because of this. But the consequences of this are not much more than the double strand break if we keep as it is. Right? So double strand break has to be repaired because double strand break can lead to various harmful cases. Okay, right? Cancer is there, or we can say cell death, right? So this is very harmful than this mutagenic process. Okay, so non-homologous enjoining is used for the repairing the double strand breaks. So here the most important proteins are the Q proteins. Q70, Q80. Most important fundamental proteins are this, which recognize the double strand break and join there. See, we can see here Q70, Q80 has joined, and they initiate the repair pathway. So next they will invite DNA picases, protein kinases. Okay, we know kinases is for the phosphorylation reaction. So DNA picases will initiate the next protein Artemis, which is a very important Artemis. Artemis is a endonuclease as well as exonuclease. It is having a nuclease activity, right? Means it will cut the strands. Okay, it is responsible for the processing of the strands. Okay, for the processing of the strands. Then the synthesis of some nucleotides take place, which is not much accurate. Okay, then the ligase 4. These are the types of ligases: XRCC4, Terminus, XLF. So these are the types of ligases. This will come and join the strands. Okay, and thus the strands are joined. So these are the very important proteins. Let's summarize. First of all, Q70, 80. Very fundamental proteins, important proteins in the non-homologous enjoining. These are coming as heterodimer. Okay, and this will recruit another proteins known as DNA picases. These are the protein kinases. Third protein. Which is the third protein? Artemis. This is a endonuclease as well as exonuclease. Right? This what they do? Processing of the strands. In processing, nucleolytic activities are there. Right? And the last protein are the ligases. We can see the types of ligases in the diagram. Right? Then what else? I will just write here processing the strand. Processing the single strands. Right? So this is a pathway of non-homologous enjoining. It is just short and sweet pathway. Only the 270, 80 are the fundamental proteins. They initiate the pathway. They recruit the second protein DNA PKC, which are the kinases. And the Artemis. Artemis is for the processing. Processing includes nuclease activity. They will cut the strands and synthesize some few nucleotides, okay, which are not accurate, right, with the help of polymerase. And then the last protein will come for the joining. Those proteins are ligases type 4 basically. Okay, so this is all about non-homologous enjoining. Homologous pathway we are discussing on the recombination video separately. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section.